Thanks for joining me. It's a lovely night and you're inside, which you shouldn't be. You should be outside. There's lots of flies, doing what flies doing, and hopefully creating some of these little things as we're going to talk about tonight. In all my enthusiasm of last year's seminar of the larval stages of Diptera, thought, oh, do you know what? A pupal stage of Diptera would be very easy. Transpired, I was hugely wrong and now quite depressed by it. But hopefully it will be okay. If you don't know me, I've been working at the Natural History Museum. I'm probably in my third instar stage right now. I definitely aren't in the pupal stage. I definitely have not reached the adult stage either. But I have been working with the fly and flea collection there for quite a while now. And I've been meeting all sorts of experts from around the world. But my specialist groups were in the larger Brachycera or lower Brachycera, and some mosquitoes and the fungus gnats. So it was a big deep dive into other groups and areas to see what they were doing when it comes to the pupal stages. So first of all, how do we get to the pupal stage? So growth is obviously a very important stage, and with most animals, most insects, there's a feeding stage is the dominant stage, and it's an important part of the individual's ontogeny, which is the development of an organism from the egg to an adult. Now, there can be subtle changes from going from an egg to an adult, or there can be very dramatic stages that take place in an insect. So they can either go through a series of instars, a series of molts to grow bigger. And this we see in the very basic ones. These are in the apterygotes, the ones without wings. So these insects that hatch from eggs, as you can see from this diagram here, and they basically look like the adults, except for they do not have any genitalia. Okay, so these will grow relatively slowly, maybe a nice lifespan between 4 to 8 years has been recorded with a lot of these. And these include things like your jumping bristletails, your silverfish, and the insects that got demoted into hexapoda, like the Clembula and Protura and Diplora and things like that. But as we move into all the pterygotes, they undergo different sort of more or less marked change in form, whether this be a metamorphic, so not complete, or a complete metamorphic, a complete change in their body plan. So we divide them up into the hemimetabula. Okay, here's an example here with an orthopteroid insect, a grasshopper. Okay, and these undergo this more or less partial or incomplete metamorphosis. So this includes most of the insects, such as the Hemiptera, the true bugs and their allies, the Orthoptera, the grasshoppers and those. We get the bark lice, we get all of those sorts of things. What we most affectionately call in the museum small orders. Okay, so these are the ones that go through this incomplete metamorphosis, and you recognize them. But then the big hitters come in, and these represent 80% of described animals on the planet. They are holometabolists. They go through a complete metamorphosis. I was just talking today, I was being interviewed by someone on BBC, and they were like talking about these alien creatures that are insects, which made me giggle, considering most of life is an insect. Most animals are insects. So really, when we're talking about aliens, we should be talking about ourselves. We're very, very dull mammals in comparison to what most of the insects, especially the flies, get up to. So these go through complete metamorphosis here. We go through the egg, the larval stage, depending on what the species is, how many of those, the pupal stage, which I'll focus on today, before we go on to the adults. Now, most people, when they think about insects, they're just thinking about adults. And actually, this has been a major stumbling block in research of today's topic, is that the pupal stage is the most ignored stage out of all the life cycle. So before we go on to talk about the immature ones, I'm going to talk about why flies are great, because I always do, because they are. And one of the reasons they are so great is that they're mega abundant. Okay, they are supremely important at getting everywhere and doing everything and looking like many different things. Okay, they are so diverse. There's over 7,000 species of flies in the UK. Hopefully all of you are members of the Dipterans Forum, which is a very pleasing society where everyone is very kind and considerate and they just want to spread their love of flies and the research. And I'll do a plea at the end because we do need more people to go out there and start exploring these hidden stages within the insect's life cycle. Very abundant. They outnumber us 17 million to 1 or something ridiculous like that. And they get everywhere. They get everywhere across the world and they get everywhere outside and inside. Half the planet is thought to be of parasites when it comes to the animals. And within those, there's a lot of flies going, yeah, we like doing this too. So they've got themselves into very strange and wonderful places. And let's talk about their life cycle. So I would have showed this series of slides before if you came to the little larvae on Lectron larvae, but it took me ages to make this, so I'm playing it again to get a double usage out of it. So yeah, the adults, the males and females get together and have some eggs, depending on how many species, what species they are. 1 egg, more eggs, lays more eggs. Oh my gosh, so many eggs. We don't know what to do with them. And depending on the fly, it's dependent on how the mother has any care. But let's take 1 egg. This will hopefully grow, develop, hatch into a larvae, and the larvae will have a lot of fun along its life stages, consuming as much food as possible. Now, this is really important because often they are putting down the reserves that need them, A, to be able to transform or to metamorphosize within the pupal, but also it will sustain the adult stage. Many of the adult diptera do not feed or will take tiny bits of nectar, sugars, or just water. So how much food they put down, how much fat, how much reserves they put down in the larval stage is very important. So the periods between each molt is called instars. The exact number of this varies. What they look like varies. This is a higher maggot. I like calling them higher maggots. You can tell because it's got reduced head capsule, lovely looking anal spiracles there. And this is very important for identification. And it transpires that in many cases as well, the final instar stage and the pupal stage will have very similar characteristics that can be used for identification with some of the species. So here we go, ta-da, the pupal stage. Okay. Arguably the dullest looking part often in the life cycle. But what I think is the most extraordinary part of the life cycle, the ability to transform, the ability to modify most of your body from one form to another and to develop so many different features along the way in this tiny, and it can sometimes only be hours that this transformation happens. Sometimes it will be many, many months, but the shortest I know of, and I'm sure there's many shorter, is about 10 hours, which is extraordinary to change your body form in that shorter period of time. And then, ta-da, they pop out. So this is a terrible video, and hopefully I'll get a link to another one later, but it shows you the emergence. This is a parasitoid as it emerges out, but within that body will be its pupal case. So I will talk about that in a bit more. So first of all, what is a pupa? So the larvae, the insect itself, the fly itself will go through 2 proper metamorphic molts. So 2 different stages where it has to get to where the adult form is. So there is a larval pupal molt. So this is your first one to get to the pupal stage, and then the pupal adult molt to allow the transformation for which the pupa it has undeveloped wings into fully formed wings. Now it's often described as a resting stage because I think this is far from accurate in describing it, because internally there's a lot going on, and with some of them, because you know that flies never read the blueprint about what they're supposed to do, it actually can be very active. And some of them can move away from situations, some of them can defend themselves, and some of them get up to some naughtiness as well. But I'll talk about this in a bit. So inside the pupal case, we have the little pupa that is developing. What is quite fun nowadays is that we have many different methods of being able to understand what's going inside the pupa without actually disturbing it. And this has really amazing applications for when it comes to things like forensic science and helping us to solve crimes, because we're better able to age what's going on whilst the insect is still developing. Before we go on to that, first of all, how on earth did this start? Now, I was a naive idiot and I never really thought about the pupal stage. It was one of those things that I'd learned, I'd accepted, and I'd moved on until I was challenged going, Erica, have you thought about it? And I was like, oh, no, I haven't. And now I have to think about it, and now my brain hurts. But I mean, I'm not alone. Aristotle was thinking about it a long time before me, 2,000 years ago, trying to understand how the larvae and pupa came about. Now, ancestral insect species didn't undergo through metamorphosis, as I've just talked about. And we know that phylogenetically, it only evolved once with a complete metamorphosis. Okay, it's not one of these things that has evolved many times and we've just got convergent evolution. So this is a paper that I've grabbed a figure from. It's a really good paper, Truman and Ridderford, published in 1999. And it is one that discusses one of the many theories, because obviously there is, on insect metamorphosis. And we start from the most primitive form, so A at the top, to basically our flies, F, there we go, at the bottom. And we've color-coded it, or they've color-coded it, so you can look with the yellow ones being the pronymphal, protolarval, larval instars. So this is how they're matching them up. With the nymphal pupal stages, the green ones. And then we look at the wing bugs, the wing imaginal disc and the wings that all correlate to each other, and they're in the purple. Now, what you've got here, it shows the possible steps from the transition from this hemimetabolist to this holometabolist life cycle. So going from this incomplete to this complete metamorphosis. And it shows it in relationship to the juvenile hormone production, which is the blue bar going across, and the growth directed towards the imaginal form, which is the black triangles. So this is trying to get it to develop. So the species with them are more ancestral, with the imaginal disc gradually developing earlier, if I've said that the correct way around. So their hypothesis, the Truman-Ridderford hypothesis, was that the evolution of the metamorphosis is based on the premise that basal insects actually have these three different life forms, this pro-nymph, nymph, and adult. And they propose this is directly comparable to the larval, pupal, and adult stages of the holometabola. So they were proposing that, that the females may have deposited their eggs in protected spaces, less vulnerable predation. The behavior would have selected for adaptations, maybe for a protolarva to burrow efficiently away from these hiding places. So you're extending this proto-larval stage, okay? And this is gradually going from proto-larvae to larvae. So these are basically a precocious egg, a walking egg is what's going on at this point. This is what they're saying. And that eventually that they have to go through this final stage, this pupal stage, to form into this adult. So all of this early bit, this larval stage, this pupal stage, are meant to be like the embryonic development. Now, obviously, they're not alone, because anyone who suggests anything, there are many theories. So these de-embryonization theories, this is just an expanded embryo, is the larval stage. There's about 3 of them. So Berliz's one was the 1913, the one in top middle, and this is the one that kind of kicked them off with this. And you can see that the Truman-Ridderford one is basically just a slight development of that one. Okay, So you've got the Harvey-Lubick one, which considered the pupal stage to be a continuation. So those three are your embryonic ones. You've then got one suggesting the pupal stage of your, this is number D, the Polyakov. I can never say that, I'm very sorry. Arose for subdivision of the imaginal stage. You've got a rather bonkers one from Heslop and Harrison, because we obviously know that the holometabolists evolved from the heavy metabolists, but this is suggesting a common ancestor of both. So I think we're quite confident we can leave that theory out. And then Hinton in 1963 considered the pupa to be homologous with the last nymphal stage. But most of the evidence is pointing towards the Burlese-Truman-Ridderford embryonic development. There is a lot of work still going on this, and I would suggest that you read Grimaldi's evolution book, as well as some other current papers coming out about it. And what's going on? How is this development happening? Well, again, this is Grimaldi, this is his new book, The Complete Insect, and I've just lifted this image straight from that. So I've probably broken a lot of copyright infringements trying to do this. Sorry, David. He's a very nice guy, so I'm sure he won't mind. So the insects, the larval stages are just there, stuffing their faces, as is the way of the larvae. And once they get a critical size, that's when they can go into the terminal growth period. They can still keep going, but it will start off the pupation regime as well, the process. So this is controlled by an insulin pathway, which it's like uses insulin like peptides and their actions to promote growth at different levels. And then you've got the ecdysone, which signals molting events. So you've got all of this endocrine regulation going on. And again, it's critical how large the insect is to how the pupil is to what the adult will be. And then what is going on inside? Well, this gets all a bit funky. And I don't know if any of you have ever read any of Swammerdam's early stuff where he cut open bees and he cut open moths and things like that and caterpillars to look at and pupal stages and chrysalis with that. Maybe not, but it's a fantastic read, and I definitely know that. Luckily, he didn't start cutting open flies, because they wouldn't have had a clue what was going on because it was so unobvious. But what is going on, and thankfully we have a lab organism in a fly, Drosophila melanogaster, and Drosophila in its kin, and there's so much research has actually gone on to tell us what's been going on. So within Metamorphosis, well, actually, let's go back a bit. So the adult features develop fully only at the end of the larval stage. Okay, so these clusters of cells, these imaginal discs, these clusters of cells will get together during the final larval stage. Now, some of them will actually start producing adult features, or some of them will wait until the pupal stage for them to do that. Now, these clusters, as I said, will undergo moderate growth during the larval stages, and their fate is already determined at this point. But in the pupal stage, they properly kick into action. Now, these different clusters of cells called imaginal discs, I'm pointing at my screen like you know what I'm talking about, but you can see they're color-coded on the screen. If you look on the right-hand side first, at the front, we find the pairs of imaginal discs for the head structures, such as the antennae, the eyes. Around the thorax, we find the leg and the wing discs and the halter, and the posterior end carries the genital disc. So during the pupal stage, the cells of the imaginal disc, they undergo this rapid proliferation. And what is quite cool is the discs invaginate, so that the center of the disc becomes the outside of the disc, which becomes the tip. So for example, on the left-hand side here, you can see this disc, and you can see that the tarsi are at the center of the disc, and by the time they've invaginated and developed, you've got now the tarsi on the outside. So this enables this growth to happen. The periphery becomes the base of the structure at this point. I just imagine this kind of like woof, as all these different features are developing within inside the puparium. And here you go, someone, that's very sad for the little fly here, but somebody has dissected all the way through. This is Drosophila guttifera. So I've put little papers in where I've got my sources from. And you can see as the larvae gradually hardened for this pupa, with, inside a puparium in this case. And so they dissected it open, and you can see as the pupae is developing how their little features are starting to come to the fore. I think it's quite adorable. We've got the wings and the eyes darkening, as you can see. Obviously, there won't be a total development of the wings till they actually leave the pupal case. Now, I got slightly sidetracked with this bit, and I've got this little book called Insect Transformations, which is lovely. It's published by the Library of Entertaining Knowledge, which is arguably one of the best things. And it was published in the 1860s. And I'm just going to read you out a little bit on the fly pupa. We've referred to many numerous species of what I'd called common flies, which is rather sad. Now, unlike most larvae, these never cast their skins, not when they change into pupa. The maggot of the common blowfly, for example, when it is to undergo its transformation, quits the carcass where it had been feeding and buries for an inch or two into the first soft earth it can meet with. Here it draws in its body into a shorter compass, and the soft skin being thus condensed, it acquires a thickness what it loses in extension. Its moisture also disappearing by evaporation, or more probably by internal absorption, it becomes hard and tough, like thin parchment, of a dull reddish-brown colour. The form is now of an oblong ball, and it was from that circumstance termed an egg by Reddy and other early naturalists, a term at which Swammerdam takes great offence in this instance. The various changes undergone in these included the insect were traced from hour to hour by Rému, with his usual patience and accuracy, but few of the minute circumstances detailed by him would probably interest our readers, except that in casting its mandibles, which were henceforth useless, they are not thrown off on the outside of the case, but remain on the inside. I love the idea that there's a man 150 years ago just looking at the inside of discarded pupal cases to see the mandibles. And just the final bit I just want to point out, he says, were such an extraordinary transformation as this to happen to one of the larger animals, it would be held forth as altogether miraculous. Were a lion or an elephant, for example, to coil itself into a ball, compressing its skin into twice the thickness and half the extent, while it remained uniform in shape without joining or openings, and at the same time, were it entirely to separate the whole body from this skin and lie within it, as a kernel does a nut or a chicken an egg, throwing off its now useless tusks into the corner, and then after a space, should it acquire wings, loving this, it's Dumbo, break through the envelope and take its flight through the air, there would be no bounds to our admiration. And that's the thing. I arguably think that what is going on in the pupal stage is one of nature's most finest things, but most people ignore it. And to be fair, most of the time it's hidden. However, now we're developing new methods in which to be able to see it. So, for example, this was carried out by some research colleagues of mine, Martin Hall and Daniel Martin Vega, along with Thomas. And this was looking at the micro-CT. So we are actually in real, looking at the development inside of a puparium within a CT scanner. Now, having been a cyclist in London, I've been in many a big CT scanner, but at the NHM, we have a little one, and we're able to undertake this research. So you can see there is the air bubble, and the air bubble gradually changes, disappears. And over time, we can see the formation of different limbs, such as the wings developing, there's the head, the thorax, the abdomen definitely coming along, the eyes, all of these sorts of processes happening. It is truly extraordinary. So what makes a pupa a dipteran pupa? So the first forms are typical, and I will be using the term Nematoceran, even though it's probably not a valid. So Nematoceran is no longer phylogenetically correct, but I will be probably slipping in and out of that term. Now, for the Nematocerans, they are defined as having an object or body-attached appendages. Okay, for example, this is the pupil of a crane fly. They have identical head, thorax, and abdomen, but covers of the antennae, the legs, the wing pads attached to the pupa, etc. So you can see quite a lot of the form, and this is very useful when it comes to identification. The second in the higher flies is exarate, i.e. the appendages are free. But you don't know that because most of them are invisible to us because they are hidden within a puparium, okay, which is this rounded, harded, panned feature of many of the higher flies. Just to be flyish about this, not there are some nematocerans that have a puparium. There are some larger Brachycera that have puparium. So there's all sorts of fun stuff that occurs throughout the order of flies. And to remind us what's going on, so here is the diptera phylogeny. This is taken from David Wiegman's paper in 2011. So we've got these 3 major episodic growth periods within flies. And basically, we've got the first type of pupa just seen in these flies. Obviously, apart from this little stratomide, throws them out. Oh, look, we've got some cessids and things down here that also have this puparium. But it is this lot that have taken the puparium and run with it. And these are split. The higher flies, they're Schizophora. Sorry, I realized you couldn't see my, the Schizophora and the Aschyzer, or the Schizophora, Aschyzer, however you want to pronounce it. It is how they leave the puparium, which is how they got their names. So let's have a little talk through some Nematoceran pupae. Okay, a lot more happens with the Nematocerans in comparison to the pupae. They're a little bit more exciting than the higher flies. It definitely has to be said. All sorts of fun things going on. And it's really nice because I've done a lot of collecting on field trips. And one of the things we can do is collect their puparium, to collect their pupal skins, because a lot of the features are still on the pupal skins. So let's remind us they're objects. So we've got the appendages and the external structures of the developing adults visible on the outside. The majority of them are immobile, but obviously these are flies, and obviously there's exceptions. And the adult fly emerges from the pupil through a straight line, this longitudinal seam on the dorsal surface. I don't know if any of you have just spent time watching mosquito larvae hatch. If you haven't, it's a lovely way of spending an evening, just watching them hatch. And you can see as they burst through, generally on the water surface, and pull themselves out through this line. Second book recommendation is obviously Flies of the British Isles. We have got the original drawings of these in the library, which is so lovely. And he does some fantastic little drawings. Hammond does the drawings, doesn't he? Where's it go? Anyway, showing the different dipterous pupae. So we've got the Nematoceran pupa here with the tipular, chironomus, simulids, and the mosquitoes. And the mosquitoes are quite fun. Moving up to the large Brachycera, where we get the reduced, the pupa, and then we got the puparium here. So let's have a look at the tipulids. Now, the final larval skin is shed to expose the pupa. So the final larval skin is what makes the puparium in the higher flies, but this is shed. This is normal for Nematocera. Now, in the crane flies, this stage can be quite short, from a few days to 2 weeks, and none of them overwinter in this stage. So a lot of flies will have an overwinter in pupal stage or a diapause, but not in the crane flies. Now, Crane flies are odd in that perhaps all develop the adult body inside the late larval phase rather than the pupal stage. It's like, oh, flies, what are you doing? Okay, so this is the premature existent is called a prefarate adult. So when the larval skin is shed to reveal the pupa, the adult basically has to then derive its air supply from the newly exposed anterior spiracles. So if you look on the right-hand side, the head stage, you can see two little spiracles sticking out, these respiratory siphons or dee boppers, you know, and they will occur a lot across all the different puparia, and some of them are very diagnostic and rather strange. Sometimes the adult females in some of the crane flies, she will actually have mature eggs as she emerges from her pupa. So they often mate immediately they hatch. So this is called pro-ovigenic. It's not very typical in Diptera. It's more typical in Hymenoptera. But the crane flies are like, right, ready, let's go, let's get it over and done with. And the adult crane fly often has a very short stage. Recommend Alan's with a lot of help with other people. Here's a book on crane flies that talks about this. So within crane flies, you've got some variety as well. You've got some aquatic adaptations. So aquatic larvae and pupae have had to go on to do different things. Many of the aquatic larvae will pupate in the sand or soil, but others stick around. So we've got an aquatic adaptation here with, this is the Eryptera swallowder, best name, okay? So Alan described, this is the Highland splay as he describes it, and I guess you can see where it is found, up in the Scottish Highlands. Very few known locations, which is sad because it sounds really quite fun. So this, the pupil can actually, it uses those respiratory horns to actually soar into the vegetation to obtain its oxygen from there. So this is quite a fun adaptation. You've got another one, which is the Pseudolimnophila locorum. And this, and I love the way Alan writes this migrates in a cunning way. So this one is able to get out of trouble if something is going for it. But what's really quite funky is that they have developed a plastron. So this plastron refers to a series of hairs or bumps on the surface of aquatic insects that helps them breathe underwater by trapping air. So this has the plastron, we know it's independently evolved twice in the beetles, but guess what? In the fly, it's evolved independently 8 times. So it's found in the crane flies, it's found in the Tanypterids, it's found in the black flies, the Simulids, the Blephoceridae, the Deuterophlebidae, the Empids, the Dollies, and it is even found in some high flies, the Canassids. So it appears, which is quite fun. So you can see here, this is a diagram on the this is actually, and they're on the gills, the spiracular gills. This is Ellipsothrix remota. And so you have these series of bumps, these series of holes along this ridge that enables this air bubble to develop. So there's quite a few species in the UK that have that. So you need to go out there and find them and image them for us. Lot. See, this is an image of one of them, the Marmorata. Okay, it's very poor images, but you can see on the respiratory horns. Okay, here we've got the spiracular gills, which is the B, you can see here at the very end. And on those, so C is a closer up look, and E and F are those plastron forming areas on those gills that are very high magnification. Cyclorrhapha. So they've got a respiratory siphon. Well, they've got 2. But one of them is rudimentary and one of them is extraordinary. I think there's no other way of describing it. Alan in his book talks about the larvae and the pupae being very distinctive. The larvae are fully aquatic and have extendable periscopic breathing tube at the hind end. Very much similar to the Aristolini, the rat-tailed maggots, and the pupa are also aquatic with this long bristle-like breathing tube at the front end, but it's only one-sided, a curious asymmetry, as he puts it. We're not quite sure why it's doing it. We don't know anything really about them, and I've seen no images, real images of what these will look like. So it's a distinctive feature, this horn. Nothing else really has a horn looking like this with it not being in a puparium. The orientation of this horn can differ between the species, and the pupal stage is typically between 5 and 12 days. Okay, so they're definitely worth going having a find to look for. The ceratobegonids, the biting midges. There's 173 species in the UK, so there's lots of activity for you to go and find these. Everybody loves the biting midge. The thing about the biting midges is they're an incredibly diverse family of flies. So there's 173 in the UK, but there's over 6,000 in the world. And they basically occur everywhere where there's a little bit of water. So Art Borkamp is one of the world experts on them. He's describes them from the tropics, high Arctic, everywhere, lots in the UK. He's done a brilliant publication. So this one here in 2014, Pupa of the Biting Midges. So you're like, brilliant, well done. But it has a warning straight away going, only 13% of all described Tanyptera are known as pupae. 13%. And this is quite a high figure, I feel, in a lot of the literature that I've come across. Okay, so most of them are only described by adults, very few larvae, and pupa are even more ignored than this. This is common throughout. But immatures are quite diverse morphologically, and they can be represented in all sorts of little aquatic margins, damp forest floors, etc. in the UK. So here's one. This is in the Forsythia minor, and this is Forsythia maya. These are terrestrial. These ones are the adults who pollinate chocolate, so most people should like these. Now they live under bark and in cavities and cow manure. Fabulous place to grow up. And the larvae pupate into when they're fine, they're often in little groups. So look, woo, they have this little circular group of larvae, and they will pupate in this little circular group. I just think it's rather nice. I think they've all got together beforehand. Now, what's quite fun is they keep their larvae exuviae on their abdomens. Okay, and these exuviae have these elongate secretory setae, which are highly sticky, and it's often repulsive to predators, or it can act as the opposite. So it acts as like a gin trap. It is a mobile spiked traps. So these things have got all sorts of things going on. So even the pupal stage is doing something. Chironomids. You didn't know, a highly diverse group. And this is quite exciting because now we're getting into the parthenogenic species. So we even have here females that are the parthenogenic females may lay eggs while still enclosed within the pupal cuticle, which is a whole lot of crazy if you ask me. So this, the pupal stage may be quite quick in these, maybe 2 days. This one here, the Paratanetasis gremi, okay, these can be a nuisance, but these are the ones that can reproduce parthenogenically, i.e. they don't need a male, it's an asexual reproduction, and it can take place within the pupal stage of the female. So she's active before she's even started, which I think is quite amazing. Mosquitoes. Now, we love a mosquito. Look at this little cutie. This is called a little tumbler. As many people, please do read some 18th century literature describing these tumbling up and down water columns. It's rather romantic. So about 35 species in the UK. And the pupal stage is the resting non-feeding phase. I mean, it is resting kind of, but it is still quite active. They are found in stagnant water, not running water. They don't have any external mouth parts at this point, and they can swim and flip by moving their abdomens. And they have these trumpets. So you see at the top of the pupae, just below the water surface, you see these trumpets, these respiratory siphons that enables them to breathe during this pupal stage. And this stage, it may be between 1 and 4. Now, what is really quite exciting again for anyone who does mosquito research, maybe we're a bit, I don't know, but you can identify many of the species based on pupal characteristics. In fact, there are some species that have only been described on their pupae. And although it's very easy, I'm sorry, this slide is a bit blurred. Yeah, with the adult mosquitoes, the larval stage of the different major groups within the mosquitoes, which you would have all heard if you came to the previous talk on mosquitoes, the pupae only differ slightly. But the anopheline pupae, these are generally larger than the aculicine pupae, and they tend to be slightly more rounder than the aculicine mosquitoes. The respiratory siphon is no, it's quite similar. Sorry. So you have quite fun stuff going on with mosquitoes. Thankfully, I spent a little bit of time in the Caribbean. I got quite obsessed by this species. It lives in crab burrows, so I had lots of fun. But what happens behind closed doors in the crab burrow was very naughty, because you get what's called pupal guarding. So this is a Dinocerathes. I can't pronounce that. Sorry, cancer. You can see where it lives in a crab burrow, can't you? And the males practice what's called pupal guarding. Now, he's quite different to most mosquitoes in that the males will listen, obviously, in the air to the wing beat vibrations of the females. But this one, he uses antennae and his tarsi to actually sex the pupae, which is quite extraordinary. And when he's found a female, he waits and he guards her. So he's hanging around on top of her, waiting for her to emerge. And then he will beat off any other male that comes close. He's like, back off. I found this lady. And then he's got this very long antennae. He's got claws on the end of his tarsi. And as she's emerging, he basically rips the pupal case and then he's on her. She has no choice in the matter. It's quite rude, really. And often she's quite receptacle. But you can find these pupal skins, which are amazing. I've got a little soft spot for Brachycerids. Okay, I know they're not UK, but they're very cute. I think they look like little, is it K9, Doctor Who? That's probably aging me of this group. But they're able to sucker on, because these live in fast-flowing streams. So these pupae underneath will little sucker on to the rocks or whatever substrate they can get on. And you can often get loads of clusters of these pupae. But we have some funky stuff living in fast-running streams. Simulids, black flies. I mean, if you haven't seen a simulid larvae, I don't know what you've been doing, but you need to go and see a simulid pupae. Now, there's about 35 UK species, and they live in the fast-flowing water where they can filter out particulate matter when they feed. And when they are fully fed, they construct this silken slipper-like cocoon, okay, on the substrate. The larvae, I'm going to actually read, this is there was an anonymous guy who posted this photo in this web link, but I'm going to directly read it out because I think the way he wrote it, he deserves to be acknowledged. So the larvae then molts into the pupa. So you've got this larvae, and the top image is the larvae. Okay, it's almost fully developed with fire adult in its pupal case with its cocoon. So the gills are sticking out. So these are lovely gills sticking out. The second is the empty pupil, and the third are the empty pupal cases. So you can really see this line, this longitudinal line, which they burst out of. But the larvae, it says it molts into this pupa within this cocoon. The pupa has a bilateral pair of cuticular projections from the front of the thorax. These spiracular gills extend outside the cocoon into the current of the stream or river and serve as respiratory organs. When the fly is fully developed within the pupal case, it generates a bubble of gas, splits open the pupal case, and is transported to the surface in this bubble, whereupon it bursts into the air like a Trident missile. Brilliant. That is probably the most dramatic birthing ever. It's absolutely fabulous. Okay, we got Cicadids. Okay, owl midges. I refuse to call them dragonflies. 101 species in the British Isles, and these are incredibly short. This is 20 to 40 hours during a pupal stage. I mean, they are like, let's get out of here, let's go. The insect doesn't feed, it stays submerged, it's still breathing through its spiracle, we can see them here, and it really quickly just changes from the larval to the adult, and it's out of there. Cessids. Oh, cessids. Where do we start with a cessidimiad? 620 species getting up to all sorts of things. Now, the larvae of gall midges, they use various strategies and adaptations to survive in unfavorable environments. Now, one of them is spinning cocoons during their life cycle. And they have these secretory glands, these salivary glands to create this cocoon. And during the larval stage, these glands gradually shrink and reduce in size. But often they were pupae within this cocoon. Now, they help, it's really good because they help prevent attacks from predators and parasitoids, changes in climate, etc. And the cocoon itself may also include bits of stones, anything they found. Now, cocoons are not just found in cessidimiads, they're also found in chironomids, caraplatids, fungus gnats, all sorts of things. But cessids not only make a cocoon, but some of them also make a puparium. Now, this is exceptional in nematocerans. And this Hessian fly, this terribly destructive fly, which you can find in the UK, is one of those puparium-making species. So here's the cocoons. This is a fungus gnat, an image of a fungus gnat within a cocoon, and all of these are cocoon-making species. So it's not just found in the chironomids, the fungus gnats, the cessids, etc., but you can find it in the higher flies and even in some house flies, the mustidae. But cessids don't just leave it there. So not only can some of them make cocoons, not only can some of them make puparium, but they also, some of them have this pediogenic process. Yeah, just like we talked about before, some of these they go through parthenogenesis within the pupa. So this brilliant paper, Pupa Pediogenesis in the Cessidimiads. Okay, this was published in the 1950, late 50s, early 60s. Okay, and I love it. This is a UK publication. You'll be glad to know you can go and find them yourself. And the author received a sample of mushrooms compost from Cornwall, and it was infested with these larvae. Now, some of them changed into normal looking larvae, which is the image 7, whereas some of them produces other sort of larvae, image 8, which were slightly oblique. And then he describes these larvae. He calls them larvae, but it turns out these were probably a pre-pupal. These were definitely a pupal stage. They molted into this unusual elliptical form, which is perhaps best regarded as a hemipupal. So within this, though, within its hemocele, it produced numerous embryos, which later emerged through the cuticle of the larvae. Now, I have this image in my head of them just exuding like immature stages of cessids here, which is quite remarkable. I don't know. So he said the smallest hemipupae recorded 2 young, but the largest one, which was 2 to 6 millimeters by 0.74 millimeters, so we're not exactly talking big, produced 71 young. Amazing. Skyarids, the dark-winged fungus gnats. Again, you've got pediogenesis going on in these, which is amazing. And you have, luckily, a really good work by Frank Menzel, Jane Smith, and the wonderful Peter Chandler And this was a publication that came out in 2006, and they were looking at the scariid fauna of the British Isles. It is hugely under-recorded. But one of the terrible things to read in this paper, I mean, they did a fantastic job. They looked at 14,987 specimens that were checked and recorded. That comprised 12,529 males, 2,419 females, adults, 34 larvae, and a whopping 5 pupae. We have a lot to learn about many of these species. So here's some fungus gnats. These are quite cool. They use the silk to which they can they produce in their larval stage, which they use in the pupal stage to hold them in place. And again, you can go online to the RES, the Royal Enstock website, and have a look at this book, the immature stages of the British Isles. It's great. And here we're moving on to the Brachycera pupa. We're getting a little bit more funky. And this one is very funky. So the Strateomides, the soldier flies, this is where they do not shed their last cuticle. So this is a convergent evolution, and they have their own type of puparium formation. So it has many of the similar characters of the larval stage and the bristles, very short breathing tubes, but it's much more rigid than the larval stage. And in some of them, in the aquatic species, they can be quite buoyant as well. So you can see them floating around. These would be great to image, everyone. Here's an exuviae of a pupa of Lafria, which I came across in Scotland. This is in the robber flies. Now, when they pupate, many of the species will go down into the soil between 1 and 10 centimeters, or they will go into rotten wood, as this one does here. And you can often find these exuviae, and again, they have got really good identification features on them, so they should always be kept. We have a lot in the collection. Now, this pupal stage for the acilids is about 2 to 6 weeks. Bee flies. The pupal stage is develops often inside the host's nest. So, for example, in the Bombylius, this is in the solitary bees or wasp nests. But many of them will just flee. So they would try and get out of the final bit of the larval stage is in there, and they would try and get out in the pupal stage. What I love is that in the ones that parasitize the osmia, the mason bees, should they actually drill its way out before emerging as an adult. So the pupa has to do some activity as well. Here we go. Here's another one. This is not a UK species, but the images were so lovely. So this is Xenox tigrinus. Okay, this is the tiger bee fly. It's found in the States. And here you were showing the hardening of the puparium. And what is lovely, all these hairs have got tiny little hooks on it. And that's the SEM. That's a high resolution image of one of the hooks on the left-hand side. And they are absolutely synchronous with their hosts. So they will wait to emerge for when their host emerges, which makes absolute sense. I'm mindful of the time. Here's a lovely one of the acrocerids. Only 3 species of acrocerid in the UK. These are the endoparasitic spider-killing flies. But I have to say that pupa seems like it's put absolutely no effort into it. Okay, it's a very small, very dull-looking pupa for what is quite an extraordinary species. But again, we have very limited information. Dolichopodids, okay, wonderful dollies. These often you've got very long dorsal prothoracic respiratory horns here and some frontal facial sutures. But they often as well spin this cocoon, as we talked about earlier, and often they will encase it with stones or tiny little soil particles in there to encapsulate the pupa. Okay, so there's lots of fun. You often miss these because you won't recognize them in the substrate. Empids. Well, funny enough, look, there's a black fly. What's going on here? What I love about empids, not only look at this hemerodromia pupae, very crazy looking pupae, but they will feed, they're very predaceous, and these feed on the larvae, the pupae of the black flies. And not only do that, the little cheeky things, they will often then use their cocoon as a little place for them to pupate themselves. So they have a nice food, and then they have a nice house created by this quite nice synergy going on. Moving on to the higher flies. So this is where we have the development of the puparium. So Cyclorrhapha literally means circular seamed, and it refers to the remarkable adaptations this group has gone to escape their hard pupal shelters. So they have this beautiful encapsulation. They're in a little bean to look after themselves. And we split them, the Aschizophora or Aschiza, and Schizophora. And Schizophora means split. And I always think of Michael Caine with blowing the bloody doors off, because they literally blow off the capsule, the head of the puparium, for them to get out of it. So here we got the Aschiza. Again, these are taken from the immature insects. These are, and some of you may recognize some of these. Phorids. Horrid phorids, only because I find their identification quite bad. Ecologically enormously diverse. There are 357 species in the UK, and luckily there's a man who's taken on the mantle of trying to help us understand them, Mark Walsh, and hopefully he'll be describing more pupae before we know it. But one of my favourite ones, and it's in this genus called Pseudactin, and this is turned up in the UK, so I'm quite pleased about it. This is one of the ant decapitating flies. So not only do they lay their larvae, they insert their eggs which hatch, and the larvae crawl through the thorax of the ant, and then before they pupate, they go into the head of the ant, eat the head inside out, and then bish bash bosh, they pupate in their head capsule. How fantastic is that? Ant decapitating flies. We then have platypesids. I love a platypesid, beautiful adult, and look at this for a pupal case. Okay, so these have got these amazing little processes, two branch processes coming all the way around. It's like a little hem going all around the pupal case. Graham Rothery, Peter and Francis Gilbert have done some on the final larvae and pupal stages of this, which is great. Often find them in the soil surrounding decaying fungus, very where the adult and the larval stages hang out. Syrphids, hoverflies, very distinctive puparium. They may take a week to several months, depending on the species. They usually pupate near the larval habitat. And if the pupa, is overwintering, it is generally really well hidden within the soil or the leaf litter. This is a rather beautiful pupa, the microdons, and these are found in ant nests. They have a really good paper here. There's detailed morphology of Microdon mutabilis. What I still think is quite amazing about this is you cannot split the adult on their identification, but you can on the larvae and the pupae. So it's showing us how important a lot of these characteristics on the pupae really are. Look at the picture on the right. That is an image from that paper of a little microdon leaving its puparium. Now, is that not really adorable? Just want you to have a look at that. Look at that little face poking through. Obviously, it's been a vicious predator eating a lot of ants, but hey, so be it. Turns into a nice pollinator. Then we move into the Schizophora. And these are quite cool. So these get divided into the acalyptrates, calyptrates, and that's based on the adult morphology. But look at the variety of form in the puparium of the acalyptrates. And the Schizophora, this is where I'm going to quickly go into this. We have to put up with a little bit of an annoying advert to start with for 20 seconds. I'll turn that down, sorry. But I wanted you to see what it looked like to have the telinium bursting out of the head of the adult. Oh, it's dinner time. I'm getting quite hungry. Right. Okay, we just have to say thank you, Tom A. Rodrigo. But here we go. As you can see, this massive airbag that expands and contracts on the head of these flies as they're leaving their puparium. And it's just a fantastic thing. And you can, I mean, you just leave out a little bit of cat food and you will find this, and it will be an amazing thing to see. And you can see them going back and forth, back and forth. And they will eventually, once the fly has emerged and has completed its final part of the metamorphosis, expanded its wings or whatever, the air sac will collapse back on its head and will result in the suture, the terlineal suture, which is the bane of anyone's life when they first started to identify flies. But it's really obvious. You can see, look at that. How on earth did that evolve? So let me get back to this. So Alex Wild, he did these amazing images of what was going on. So just to quickly pick through them, I want to highlight some books here for everyone to go away and read. So this is the Loncaids, Ian and Graham. Graham Rothery obviously is the king of immature stages in the UK. He's done a lot on them. And he wrote about them being about these puparium, talks about their colours and whatever briefly in this book. We've got some, I quite like these, chlorophids. This is quite a cute one. These can be pest species, so it's quite important that we understand what's going on. Again, huge ecological diversity though, and parasites and predators. So we need to know more. There is a recording scheme, and apparently there is a book coming. Till we see it, it ain't happening. And then Drosophila. So we talk about Drosophila a lot. We understand most of the immature, what goes on within the puparium and whatever is looking at Drosophila development, apart from the forensically important flies. But I wanted you to see this SEM of the respiratory, here the spiracles. Are they looking amazing? So you can really see here the gill slits to enable the oxygen to go into the puparium. Acromyzids. So in most cases, the larval leaves the mine when it's about to pupate, but sometimes it can get trapped in there, so you can often see it. It does not often mean it's going to be death. They can survive this, so this is quite important. There is a recording scheme. I've put it here, the Acromyzidae recording scheme. And if they do pupate within the mines, it's often on the underside of the leaf. Colour is often diagnostic in these species. Getting into the clitorates. These are the chunky boys and girls. And we've got some chunky pupae. So this is in the Mussidae. This is the hornfly, Hematobia irritans. It's really economically important pest of cattle locally. I know it's found to Americas, Europe, Asia, Africa. Oh, it's got everywhere. What is quite interesting about these, along with many of the other diptera, is they can go into diapause. Now, diapause can happen at any stage within the insect's life cycle, within a dipteran life cycle, if they undertake it. But these ones, it's specifically the pupa that goes into diapause. So they're not alone. So similar species. There's another one, sarcophagids, and there's tachynids that also. One of the ones I love about the tachynid, this is in the Exorista civilis. It's not a UK species. I don't know how it happens to the UK species. Is they actually go into diapause within their insect host. The laziness of that, I think is brilliant. So, yes, you get diapause going on, pupal diapause across diptera. Funny enough, not in mosquitoes. Mosquitoes will diapause at every other stage, but not in the puparium. Botflies, love a botfly. There is a recording scheme for botflies. Funny enough, because considering where most of them, the larvae, live, we don't, it's quite difficult often to get the puparium people. I wanted to get the larvae and the pupa of the rhino botfly, but that is far too an extreme habitat, waiting for a rhino to defecate for the NHM to agree to it. A way you can do it in the UK is we can look at the gastrophilus. Then there's that. These are the horse botflies. I put it horse boy, sorry, bot. Okay. So these are parasites in horses and other equidae. And so the female lays 500 eggs, very high attrition, and they pass out in the feces. So all you do is basically to hang around some good fresh feces to see the final larval stage to collect the pupa. Sure, that'll be fine. Absolutely fine. Summer holidays are beckoning for everyone. Calophorids. Now, calophorids are a part of the group that's forensically important. And so actually more attention has been paid attention to a lot of the features of these puparia. Typically, though, they're quite similar in appearance, light brown to dark brown. But we can now start doing all sorts of fun stuff with these when it comes to internal X-ray imaging, micro-CT scanning, and clearing techniques. So we can clear most of the chitinized area of the puparium to actually look what's going on inside. We've got the Nyctera beards. These are the bat flies. I love a Nyctera beard, and please do go and try and find these. Look here, this was found on a parish church. This is a pupa that had been stuck to the side of a parish church. I need all of you to go around roaming to looking for these tiny. It is very small, I'm not going to lie. But we only have 3 species in the UK, and I'm running a recording scheme from them, and my activity for a recording scheme is very low because of the difficulty in finding these. But the more we do. Now, what is amazing about these is that the larvae is released as a pre-pupa. So the mother, basically, she's got pregnant. The larvae is internal. She will rear it internally. She's got internal lactating glands. She feeds it internally. And just before it pupates is when she gives birth to it. Now, the actual release of this birthing may only take 2 to 3 minutes, which I think many a mammal will be very grateful for. And the transformation into a pupa will take maybe as little as an hour. It's very, very quick. Now, this pupal stage, it can be, say, maybe 20 to 30 days, but they can also overwinter to be a lot longer. It depends. So that's it. But I've rushed through, sorry, I was a bit mindful of time. I sped up and slowed down. There is much we haven't learned. And I just want to say that the final quote is this, I'm going to say this. The pupal stage is often neglected in descriptions of the immature stages, unless it is a stage most frequently found, such as the psyllids or sphaerocerids, or of taxonomic economic importance, the cheilids, the simulids, and the chironomids. For the rest, for the other 7,000 species in the UK, they are the least known stage. They are the least described. We have amazing bits of technology called phones nowadays with cameras on. We need to collect these and use them. We are using novel techniques nowadays to be able to speciate things, and hopefully soon it'll be cheap enough for us to start speciating diptera on their pupal cases. So first, I'd like to say thanks to Matt Bertoni for most of the images, all the recording schemes, the dipterans forum, and obviously the flies, the pupal stage for doing this. Thank you.
Thank you very much indeed, Erica. That was absolutely fascinating, and I appreciate the enormous amount of research and effort that would have taken you to put together for tonight.
Yeah,
really.
well, it's been fun. I've learned everything and nothing, I feel. I realized how much we have to learn. It's been quite like eye-opening. And I know there's a lot of people in the UK, so we link rear mosquitoes. We have the larval skins, the pupal skins. And I know there's a lot of people who are doing similar things with Stratomyres. Judy is a wonderful woman who's looking at, and she's like rearing things from mushrooms or ponds and things like that. So we need more people to do all this rearing, or to at least to kind of go into different habitats and like squirm around, dig your fingers in the soil, play with poo, to find all these different stages to help us put these missing parts together.
Absolutely. There's just an incredible amount still unknown, it seems. But I mean, it's good to hear of so much research is also going on at the same time with novel techniques are kind of revealing. I mean, I love that head capsule expanding. Wow, what a way to, well, perfect way. Pressure, push, crack something with pressure.
I mean, how did
Um,
that evolve? That's the number one. And then B, it's like we talk about so going abroad and looking at nature, and it's like this is truly the most extraordinary thing that happens on the planet. This metamorphosis, this idea of reorganizing your body tissue, and then boom, blowing up your head. It's just like the final like icing on this cake. It's like, and you can hold it. I made my civilian partner, bless him, sit in the garden for 20 minutes watching a fly come out of a pupal case and watching that process, and he was gobsmacked.
Yeah, I bet. And you've given us so many really fascinating anecdotes tonight. And there's so much, there's probably loads of other weird stuff that goes on,
Oh,
which we're not even going to discover for another 50 years. Um, but brilliant. I really enjoyed your Victorian description of pupation, actually.
I
It was
know.
kind of a call
I
to action speech. It felt like Winston Churchill's it was good.
well, I kept getting myself sidetracked by reading all these things, going, "No, this is
Yeah.
not helpful. This is not helpful," but they're so beautiful. And it's like, we, in many ways, we're so clinical about our appreciation of nature, and we forget to just take a step back and go, "This is amazing. This is, we really have something amazing on our doorsteps." And the Victorians, albeit they're very bad at many things, I'm not going to start on that. They did know how to write a good book.
I did know that. Yeah, no, I love reading back through old natural history journals, etc. Um, okay, well, I wonder if are you happy to take any questions?
Yeah.
Um, brilliant. Okay, well, if anyone's got any questions, they can put them into the chat, or they can put their hand up, or I don't mind if you just want to unmute yourself and say your question. I'm sure you won't be all talking over each other. It's fine. Yeah.
Silence them.
Silence. Okay.
Yeah.
Oh,
Hi.
here we go.
Hi. Erica.
Hello.
Yeah.
Hi, hello.
Yeah. Erica, I'm Prabhu. Just I would like to know if I would like to study particularly cephalopharyngeal skeleton and also anterior and posterior spiracles, how to make the slides of the pupae.
Oh, well, it depends on the species. So I'm guessing you're dealing with higher flies with the cephalorhyngeals. So you have to, with that, I mean, within the pupal case, um, I recommend that you, first of all, go to the California recording scheme or the forensic entomological recording schemes. They will show you how they do their dissections. I know that the work I showed you there, Daniel Martin Vega, he did some micro CT scanning for a lot of the higher flies, and they exposed the cephalorhyngeal skeleton. So I think so you can do it that way, or if you want to make slides, you still have quite a bit of mush, I guess, and you could pull out I'm not even sure when it develops in the higher flies. They're not my groups. I'm not being very helpful here. Yeah, I think you're going to have to look that up. I know, well, the cephalorhyngeal skeleton should be there in the pupa. You should probably be able to just pick it out, shouldn't you? I don't know.
Yeah, maybe we can put it in KOH. Is it possible to
Oh yeah,
remove?
KOH always. Yeah, but I would be careful if you put too much KOH, because it would be such a soft stage that it depends how developed the features are. But yeah, I mean, we use KOH for all the larval stages anyway, so it shouldn't be a problem. I realize you're there. You're just very dark.
Yeah.
Hi.
Yeah. I met
It's
in fly school actually. Yeah.
I thought I recognized you. Hello.
Yeah. Hello. Hello. Yeah.
If you want, you can, because I'm not the expert on this, but one of my colleagues, she works a lot on it. And I also, obviously, Daniel and Martin, they would be very up for telling you how they do it, because I don't know. You do medical diptera, don't you? So they would be the ones to speak to in this area.
Okay, thank you.
Thank you. Thanks for your question. And there's been quite a few technical words this evening that I'm also
Sorry.
quite no, it's always good. It's yeah, love to learn, love to.
There's a lot, and when I was trying to get my head around the bit of the insect development and the pupal development, that took me a long time, and I'm sorry if I still didn't manage to get it nice and smooth. It is a technical area, and there's a lot of hypotheses rather than
Absolutely,
facts.
it is all very technical. I think a lot of recorders actually don't think about pupa, which is one great thing that we're doing that you've done tonight for us, but also really don't think that much about the biology probably, and really quite focused on identification and recording, and we need to do more really about biology and ecology.
Yeah, because the biology helps us with the identification, or the identification helps us with the biology.
Yeah.
You know, so that cephalorhyngeal skeleton, that is the mouth hooks of the higher flies. And we know that he just imagine this hook and it comes out and it shreds flesh. So we kind of know what they're doing, but when the in the larval stages. So we can look at the mouth parts of the larval stages and we can kind of get an idea how they feed and what they're doing. I just like the idea of these mandibles being left behind in this puparium. I've never even thought about things like that before. It's like, oh yeah, because I mean, why they don't just get dissolved, I don't know. But in studying these, we can learn more about, in studying all these different features, we can know more about its ecology and its biology and everything and how it interconnects.
Yeah.
Because it's all very well having these names, but if we don't know what they're doing, how do we connect properly with them all?
Yeah, it's that deeper understanding really about what's going on. I was interested also, Tom, I remember Tom Dallymore, he used to, until very recently, was coming into the museum a lot, and so we'd hear a lot about mosquitoes. He's told us a lot about them, but never about pupal guarding. Which was,
there's all
that
sorts
was
of
fascinating as well.
Yeah, no, and they're not the only ones that do it. There is a lot of males will emerge before females across many of the flies. And it's just like, oh mate, give her a breather. Let her come out. So a lot of them, the pupil guarding, but often he will just rip her trumpets apart. It sounds really bad, doesn't it? So he's on the water surface, he just rips the trumpets apart and kind of pulls her out, and it's like, whoa. So she's like being yanked out of her pupil case. But
does it take them a little while to work? So they are sexing the pupae. Do they go and check individual? What does it? Or
how does it do this?
Do it? Yeah.
So when they're flying, adults, this is, when the adults are flying, the males can listen to the females because she flies at a lower pace than him. So she vibrates at a different noise. And then he will just like kind of tune in. He will slow down so his wings vibrate at the same. But these guys, they listen to the vibrations. They can sex the pupa.
Based on the vibrations underwater. Wow.
Again, I'm like, hold on, nature. What are you doing?
Wow. Brilliant.
And there's still so much to learn. I mean, I'm giving you 50 examples or whatever, even less than that. And can you imagine what's going on with all these other cases? So aquatic pupa seem to be way more fun. Terrestrial pupa just kind of hunker down and that's it. They're in a nice little home. They're snuggle in their caravan. They've got everything they need, everything. Whereas, aquatic pupa, they're intense They're like, anything still goes on. It's a crazy world out there. So it's quite interesting to, and we know a lot more about the aquatic larvae. So we think we got about 40% of the, the larvae in the UK described. So it shouldn't be that hard to kind of keep adding the pupal stages to many of these.
And the majority of fly larvae are aquatic, right? No,
No,
if you probably told us
no.
last time. But,
Anytime you feel confident in an answer, it's going to be wrong. It's just the way it is.
but the larvae that are aquatic, then what percentage of those then have a pupa that is aquatic, or do most of them?
Can't answer that.
Yeah.
So we know that a lot of them are aquatic pupae. We looked at the smulids, but these are the medically important ones, so we pay a lot of attention to that. So some of the tepulids, they may have aquatic larvae and then terrestrial pupa. So a lot of them will get out of there and pupate in a different region. But I don't know. You tell me, mate.
Yeah. In a few lifetimes, I think.
Exactly. The more you know, the
Yeah.
less you know, isn't it?
Yeah.
It's like, oh.
Okay. Has anybody got any final questions for Erica? Lots of very nice messages coming through. Okay. All right. Well, I think we'll let you go then, Erica. But yeah, it's been a really brilliant evening to hear. It's probably a unique, very unique
I
talk
know.
you've given us this evening.
I hope it was okay. Sorry.
No, it was really quite a eye opener in many ways. Things that I've never seen or heard about, or so much learnt probably by everyone, I imagine. So we really appreciate it.
No problems. Right.
All right.
Thank you, everyone. Thank you very much for the kind comments. Okay, take care.
All right. Thanks very much.
Cheers. Bye.
Bye.